Our next speaker is Dr. Fathi Karouia. He is a senior research scientist at NASA, and he is an astrobiologist. He is interested in molecular and cellular mechanisms associated with space adaptation. His team develops countermeasures to sustain human spaceflight and space explorations. Today, he is going to talk about challenges in biotechnology applications in space.
All right, good morning, everyone. I just want to thank the co-organizer for having me over, in particular Dr. Kumar and Dr. Sin, and obviously also the Indian Institute of Science. And so obviously it's a great pleasure for me to be among you, and I look forward for interesting conversation and interactions with students, researchers, and faculty members. As you will see, I come from NASA Ames Research Center, which is one of the centers in the US. And I do a lot of things, and I wear different type of hats. And today, for today's call and conversation with all of you, I will be mainly focusing on two aspects. One of which is based on my conversation with Karouia Venkateswaran yesterday, and he inquired that I focus more on flight implementation aspects. And so half of my presentation will be addressing that. And then the other half, as you can see, based on my title, would be more talking about space biotechnology. That being said, as I mentioned, I am part of NASA, but I don't speak for NASA, and I just wanted to kind of disclose that. Similarly, I'm affiliated with different organizations, some of which are non-profit, others which are for-profit, and that enables me actually to be able to collaborate and interact with people all around the world. And lastly, I'm here mainly because I've been actually for the past few years and up until recently the portfolio lead scientist for space biology. In that capacity, I oversaw over 35 flight experiments which were successfully implemented in space, 18 of which was as a project scientist and a mission scientist. And so I wanted to kind of bring back that type of experience to the audience. So I will be talking a little bit about what we've been doing on board the International Space Station, and then, as I mentioned, transition quickly to about and talk more specifically about space biology in general, and what is the flow process for us to select an investigation, and what are the different steps and gates going from an idea to something that we implement in space. And then again, I will be transitioning to kind of current and past and future capabilities for conducting space life science, and then some considerations. So we have been having crew members on board the International Space Station for over 22 years now, and we have conducted over 3,300 experiments. As you can see from this particular graph, there have been over 2,500 publications that correspond to those type of investigation. And roughly 20 to 25% of them were focusing on human health, and the remaining on space biology. As you can see from the graph here, that correspond to publication on top 100 journals based on impact factor. And as you can see, the trends, which is increasing at an exponential rate. And again, a big portion is related to human spaceflight and astronauts' health, but also a big portion related to space biology. So currently we're living under exciting times. However, I just wanted to kind of shift gears and to kind of show you how we've been successful in implementing all those different flight experiments and missions from a space biology perspective. So as you can imagine, everything starts at the headquarter level. There's a solicitations, people apply for a research, and then there's a peer review process. Subsequent to that, there is, at our center, which is NASA Ames, there is a technical review which is done. And so the idea there is that despite the investigation is receive a good score from a peer review process, is this something that we can implement? Is this something feasible? And that goes back to headquarters, and then they make a decision. And then we go to the next phase, which is phase number 2. So phase number 2 is working with a principal investigator hand to hand, and to we try to better understand what the principal investigator wants to do. And then it is the responsibility of the program and the project scientist or mission scientist to kind of work hand to hand, either with NASA as a payload developer or with a commercial partner, to try to walk through the principal investigation team with all the different gates and steps to be able to do so. So one thing is that early on we try to better understand the science and what is the plan moving forward. So from a schedule point of view, from a hardware point of view, and also from a budget point of view. And then we have a compliance review to try to kind of better understand if everything is set and we can move forward. Subsequent to that, there is a need to define requirements early on, and requirements as per samples, requirements for resources, and so forth. And during that time, you perform some testing. So testing in regards to your samples, type of media, type of preservatives, and so forth. But also a key element, which is what we call biocompatibility, which is you test the type of hardware that you will be using to conduct your investigation. And so this is what we call science verification tests. So those are a large subset of tests which are conducted and are needed for us to move forward. And then at the end of that, we have what we call a ERD review, and so an EVT preliminary review. And so the idea there is that have we conducted all the necessary tests for us to move to full scale of testing, and which is what we call the experiment verification test. And so if we have a goal, then we conduct a mock-up of the flight experiment on the ground, and then we have to show that all the success criteria are met prior to be able to move forward. So again, it's the complex chart kind of giving you a different type of flavors. So here in the middle is what I just mentioned, but obviously there's a lot of teams which are involved, and because there's a lot of paperwork that needs to be done from a safety perspective and the verifications and so forth. And on the bottom part is the science-related aspects. So again, going back to those questions, so early on you need to do those science verification tests. What are the optimal conditions for your samples to be able to grow, and then perform all the tests with the hardware that you're planning to use, and then perform the operations. And then at times there are hardware that exists, or there's new hardware which needs to be developed, and here I'm listing some of the requirements which are needed for you to move forward. So here are maybe some things to consider. I mean, because some of you are currently working on some flight project, or maybe you have, maybe later on you might want to apply for some of those. So I think it's critical and important to have someone which is neutral between the teams. And so for us, we have flight project scientists or mission scientists, and those are in some aspects the ambassador for the science, but they remain neutral. And so, because on one hand you have a science team which wants to do always more, and on the other hand you have a commercial partner which, obviously is there for profit. And so you need to have someone between those two and kind of, one, understand what needs to be done, two, be there on behalf of NASA, and three, make sure that we are doing good science. Similarly, you need to have clear roles and responsibility with all the interested parties, because that could create some issues. NO3, no good. I mean, as I mentioned, I've been involved with 35 flight experiments, and zero of them went as planned. So having NO3, it's suicidal, because from a statistic perspective, if one sample doesn't work, then what? That, you end up with NO2. So certainly you need to have more replicates to be sure that you're bringing back meaningful science. And then this is critical. I mean, test what you fly and fly what you tested. Again, I have a long list of examples where we try to kind of, have a quick turnaround and do our best. And we did some modification last minute, and we thought it would work, and actually didn't work. So that's a critical step. Then also establish clear success criteria from a scientific perspective. I think that's very helpful, because as I mentioned, things will not go as planned, and then you can go back to the program and say, okay, here are my success criteria, those are not met or partially met, and therefore you have strong foundation to be able to move forward and inquire for another flight. Again, plan for the worst and hope for the best. So always try to have cards in your back pocket, because, let's say you request a return of -80, well, at times that doesn't work. So if prior, maybe you plan for the worst and you say, okay, if we don't have -80 capabilities, maybe We might have minus 40, we might have minus 20. But is that from a science perspective feasible? You need to do those tests prior and having those things in your back pocket in order for you to kind of plan for, the worst case scenario. And then, you need to stand your grounds as well. I mean, we're all scientists, and if you have garbage in, you have garbage out. So stand your grounds when that is needed. And at times, things are not feasible. At times, there's delay, but this is part of science. And then, obviously, in situ analysis are needed. I mean, currently, we're just exposing, we're bringing back to the ground. This is like a 5-year cycles. This is no good. So now we need talented scientists like you capable of performing those and developing those instruments so we can do analysis in flight. And so here is a chart that kind of describes some of the things in regards to the current capabilities. And so the first 3 elements address the ISS, CubeSats, and free flyers. And this is things which are coming online. So from the ISS perspective, obviously, it could be automated, or there's crew which are obviously on board the International Space Station. Similarly, I mean, this is, as Dave mentioned, smaller in size, can fit nicely into those racks on the ISS, and you can, change them. From a CubeSat perspective, obviously, you have a most diverse type of environment that you can target, and higher exposure to radiation. And so certainly, this is exciting for many people, like Venkat, for example, from an astrobiological perspective. And then you have larger free flyers when you can have actually not just microbes, but also animals. And again, I mean, we're moving forward with the Artemis missions, with one having an habitat where you can expose things, and then obviously we have also some activity on the lunar surface. So here's a chart that kind of lay out what we know and what we don't know. And as you can see, the unknown is large, maybe not as large as a genomic data that Nikos was mentioning earlier. But nevertheless, I mean, we've been on the station for up to a little bit over 1 year. We've been on the moon for, 10 days, 2 weeks max. And so now, we're planning to kind of go back to the moon, but for much longer. And then one day, hopefully, we're planning to go to Mars. And as you can see, there's a lot of things which you don't know. And so how do we plan for that? And so certainly the central dogma of space, of biology, actually, it's kind of a flow process and looking at genetic information. And then you can have all those different type of compounds and molecules that you can look at using all those different platform and technology. So obviously sequencing, a microarray, here looking at the proteome, metabolome, and so forth. And then when you add all those type of information, you can discriminate, a healthy individual from someone which is not healthy. So now I call it the lag phase of space research. So currently on board the International Space Station is mainly devoted to instrumentation to kind of grow things. There is a little bit amount of analytical power, however, it's tiny. And so that's what I call the lag phase of space research, because many of those instruments are just Earth-based instruments which were sent to space. However, some of them, for example, a PCR instrument flew to the station in 2016. That instrument had been on the ground for 16 years. So that instrument was commercially available for 16 years. 16 years later, it was on the station. When the instrument was on the station, a few months later, the company decided to remove the instruments from the market. So here you end up having an instrument in space which actually doesn't exist anymore on Earth. And so, as you can see, we need to be better and come up with new ways and new type of instrumentation. And so, very early on, I realized there were some issues from a management perspective, but also issues from a scientific perspective. And so, we come up with a review a few years back to kind of specifically address that, because we had some ideas. However, we were always struggling when we were talking with NASA and NASA manager, and I quickly realized that actually they don't understand what we're trying to do, which is omics in space. So that particular review was specifically addressing that point, trying to explain to stakeholder what omics are, what are the different methods and techniques, and then also trying to lay out some foundation about, what are maybe type of technology where we need to focus on. And so please, if you don't have access to the paper, ask me, I would be happy to kind of send you. And so there was two things that I wanted to address from omics in space, one of which is not part of the paper because it came online later on, but I wanted to acknowledge that, which is something that Katsuri Venkateswaran has been involved. And the other one is the one that I've been involved, which was initially specifically devoted for nanosatellite technology. And actually, we do have also a design for the ISS. So I will refer you to that. But in particular, I want to show you the device. Again, it's specifically devoted for nanotech. 10 by 10 by 10 fully integrated going from one hand to the other. So here what's not showing is the bags where the cells will be growing. Initially we did some tests with cyanobacteria. Here you have the self-filtration and lysis system, which actually enable you to have the cells going through the system, lyse the cells using diamond dust, and then you have a silica-based filter where you can trap nucleic acid and RNA in particular. And then you flush the system with different type of reagents, and then here you have application chamber where you have a microarray. And so here you have a system which was developed as a prototype for astrobiology, but certainly we do have some ideas and some kind of related application also for space biology and the International Space Station in particular. And certainly we're looking at collaboration if there's some interest there. So again, from a technology perspective, on one hand, it's like, you know, you have a commercial-based platform, and so the good thing about that is that that would be advanced tech, current tech. And however, from a space perspective, you know, you might need some automation, and similarly, maybe the device is not adaptable to space condition. On the other hand, if from the beginning you decide to develop a space-based platform, so certainly that thing will be designed from the get-go to be able to meet and all those constraints and requirements related to the space environment. On the other hand, that will require a long time for development and testing the technologies and so forth. And then you might have something between the two, which is hybrid, and maybe have a type of kind of plug and play where maybe you can take something from the commercial available, maybe do some quick optimizations, maybe some miniaturization, and therefore you can implement something that kind of combine both worlds. So again, conclusion, I think that within the next few years we can have a large suite of platforms in space, not just going from genomics and, for example, the mini-ion, but also looking at proteomics in particular. And I think this is critical because currently, as I say, this is a 5-year cycle, this is way too long, and we need to certainly implement capabilities where we're able to perform and analyze samples in space, and then you are able to refine your hypothesis, and then you can have another round of experiments and so forth. And I think by shortcutting that life cycles, not from 5 years, but maybe from 1, 2, or 3 years max, then that will lead to, you know, more discoveries and breakthroughs. And so certainly not just for the health of the astronauts, but also for people here on the ground. As you can see, I am ready to become a part of the Indian crew selection. So that was a pictures taken in Paris, and this is the ISRO version of a spacesuit, and this is me inside. So as you can see, I'm all geared up to kind of join you and to be a part of that journey. But certainly, I'm happy to answer any type of questions, and I'm very active in social media, so you can look for me and always post for stuff related to papers, related to pictures from space and so forth. So I look forward to be able to interact with all of you later on. Thank you again.
Thank you, Dr. Karouia. We have time for 2 questions. Yes. Okay. Yeah, go ahead.
So in one of your slides, you showed worst case scenarios, right? So it's a thought experiment. At which the worst case scenarios, which doesn't usually occur, it's a thought experiment. You think of the worst case.
Right, as you implement an experiment, yeah.
Yeah. To which level do you go and stop? Because it's a thought. So.
I mean, stop the experiment, you mean?
For how many different scenarios do you think about and plan ahead?
Oh, I mean, more the merrier.
How does that process go about in your NASA headquarters?
More the merrier. So as part of the experiment requirement documents, at the end, we try to add a table of science laws. And so we kind of mock up worst case scenarios by, you know, power issues, the spacecraft blow up, there's an issues with resources and all that, and then we estimate what would be the impact from a scientific perspective, and we provide an estimate, because what would happen, the program will come to you and say, hey, we have a problem, if we do this and that, what would be the outcome? So you have to be ready by providing some information, because then there will be some conversation from a priority perspective, because you're competing among, you know, other principal investigators, and you're part of the program, and there's other program which are also competing for resources. So by providing what would be the science loss, you provide, let's say, okay, if you don't do this, then maybe 50% of the science will be lost. And based on those considerations, then the program will decide what to do next. But again, it's not just you, it's everybody is fighting for access to crew time, access to resources and all that. So more testing you perform in front hand, more options you have that you have validated, the better.
Module that you should, is it meant for ISS, or do you think it works on like CubeSat kind of systems, or are there any challenges there?
So I think I did a poor job because it actually was specifically designed for nanosat, but we have also a design for the International Space Station. So again, it was 10 by 10 by 10, so it was specifically devoted for nanosatellites.
Okay. Thank you, Dr. Karouia.
Thanks.